So it was, well, examples of the work we are doing on apple, but we are doing about the same work on peach progenies. For peach, we are working on a second generation of this cross, Fer-Jaloux cross Fantasia. And in this second generation, we are working on 200 individuals. We are also working on a backcross second generation of backcross, a cross between Summergran and another species different from Prunus persica, Prunus davidiana. And we have here on this population, backcross 2, high segregation of the genes of interest. About apricot, the same strategy, you know, with the two progeny, Coleridge cross Monici, worked in Avignon. This one worked in Bologna, in Italy, between Lito and this accession. And you know, you have here 120 individuals in both progenies. And we are working on fruit color, fruit weight, fruit firmness and texture, fruit maturation, fruit acidity and sugars, you know, to give you examples. So, as I said, first step is fruit phenotyping. So, for example, look at color. You have here in this progeny, you have here the different colors from yellow to red. And here, the number of seedlings in the different class, you know. So you have the parent, Goldrich, Monici, and this study is for, well, the results are from 2 years of studies, and you have the distribution of the seedlings compared to the parent in this graph. So the two apricot progenies have been fully phenotyped for fruit quality during 2 following years for each physical and chemical trait. And we did about the same for apple and peach. So the result is that at the same time we are doing genotyping, and you have here the 8 chromosomes, 1, 2, 3, 4, 5, 6, 7, 8, the 8 chromosomes of the basic set of Prunus. And as an example, you have here the location for this progeny of the genes controlling, you know, sugar. You have here, in the same progeny, the genes controlling acidity. So this is this type of work we are doing to locate the genes which have an interest for fruit quality. And the progress we are doing is to apply what we call the marker-assisted selection. So briefly, I will explain to you what is it. The first step is to do genetic mapping, what I explained just now. How many genes control the genetic variation of the trait? What are the effects of these genes? Are quantitative, qualitative, strong, weak, additive, synergic? Where are they located on the chromosomes? You know, you have, for example, here on chromosome 1, this gene, which is a qualitative gene, located here, is a gene for scab resistance. And we have in other places, for example, in chromosome 4, quantitative genes, what we call QTL, quantitative thread loci, which is located, for example, here. So the second step is to combine the genes by crosses, you know. So we have in the progeny different individuals, and we have the possibility, thanks to the genotyping work, to apply markers to look, you know, with the markers which are sort of flags, which indicate the presence of the genes. We have the possibility to select such individual, for example, this one, with the right marker indicating the presence of the genes of interest. So the first step is to do this work of pre-selection, and we have, of course, to validate in the field. But this technology is very interesting to improve breeding efficiency. This is our aim, to breed with more efficiency new varieties. This is especially for traits difficult to score. So the marker-assisted selection allows to monitor which genes are selected or combined, and allow to trace the genes over several generations. And this technology permits an early selection for adult traits, because this technology is applied on seedlings in the greenhouse. The second group of the second part of our work inside Pillar 6 is to develop a safe and efficient transgenic techniques applicable to sustainable quality fruit production. So you know that gene transfer in fruit trees is, of course, an excellent tool for gene function studies. There is many promising research to improve key traits on fruit trees with gene transfer, but as you know, the main bottleneck is the lack of public acceptance of genetically modified fruit. So we need methodological studies of innovative gene transfer strategies for fruit trees, and this work is undertaken on apple and pear. So we are looking for alternative transformation method without antibiotics, which is a main criticism for the previous organism, especially on corn. And we are looking for plant promoters that can regulate transgene expression in a targeted way. Also, tools for clean gene integration or silencing technologies. So, very quickly, to illustrate this work, first principle, we have to generate what we call a marker-free genetically modified plants in which undesired genes sequences can be actively removed when we want to remove. So you have here in this construct, we are in the chromosomes, we have a gene of interest, for example, for resistance to diseases. We have here what we call the marker genes, which are the possibility to select the gene of interest thanks to an antibiotic resistance. And we are working on this technology to include this gene of recombinase, and by a specialized technology, we have the possibility now to remove all this part of the construct to maintain only the gene of interest. So it will be, so this is a new technology, but we demonstrate that we have the possibility to introduce such gene of interest in the cultivar Gala.